Good evening, Professor. This is my personal timetable. So, here we have planned in this week to present you the research demo tomorrow and simultaneously also modify or to get more efficient content in our coronavirus biosensor research paper, and also working on the RF review paper to modify the content and also set in the reference in the sensor format, and also simultaneously discuss with the Jacob Shubho that about the AO biosensor research. So here we have the two titles. So the first one I think is this title goes from our side, and the second one is from Professor Ajit. So we have the two titles. So we finalize until these things, and so our basic coronavirus biosensing research recipe we have the here D5 chip, and we follow the piranha cleaning for 15 minutes at 80 degrees Celsius, and then washing with the DI water and ethanol, and then immobilization of graphene oxide on the surface. That will be done by the spin coating method, and then after drying, because we have to after the immobilization of graphene oxide, we put on the hot plate for 1 hour, is a method called annealing method. So we have to wait for 1 hour until dry, and then we immobilize EDC NHS, also spin coating, but in our case we follow the drop casting method after the graphene oxide immobilization. So we simply drop cast on the surface of EDC EDC and NHS, and then wait for the 1 hour, or it depends to dry, because we have wait for the 3 to 4 hours, and then we mobilize the antibody, and then further we use BSA for 15 minutes at room temperature, and then washing with the PBS, and then washing by DI, and then antigen immobilization and different kind of 5 different kind of concentration. So here we have checked this kind of recipe. After this, we measured LCR. We measured the capacitance by using LCR meter. So this is the table of content that we have followed. First of all, we settle down the abstract and then introduction part. Basically, we have the several kind of characterization. So we have put all the information about the characterizations in the result section, and then we discuss about our results, and then experimental section, like what we follow the basic steps and what are the chemicals and equipments we are using in this research, and then we have just describably about the surface characterization using the SEM, AFM, FTIR, and water contact angle measurement as well. And for last but not least, the conclusion and the future perspective, the acknowledgements, conflict of interest, and the references. So we follow this scenario in terms of biosensor and bioelectronic journal, and set we already set the paper in this journal format. And so this is our abstract that I just want to share with you to get your comments in the meantime. So here we focused the main positive things that we have is first is reusable capacitive immunosensor. So this is the positive factor that I'm talking about, the reusable factor, and here we have the graphene oxide. So graphene oxide modified double interdigitated capacitive, and then to having the, because we have the femtomolar selectivity within 3 seconds and a wide detection range, low limit of detection, high sensitivity, as well as the faster response that we call in 3 seconds. And the high sensitivity, as you know, is 301.6 nanofarad milli per milligram, and the sensing performance is very tremendous feature, and the scale up ability as well, and easy portability, because in our case we don't need so much heavy instruments. We just have our chip and LCR meter. It's a very easy to relocate or to get through this instrument to anywhere, and this is a positive thing that we get the result in 3 seconds. So this is the basic thing that we have in our research. And also it's a basically point of care application method. So its application is point of care testing applications, and also informatics can be useful to manage this pandemic in a personalized manner. So as in the last meeting, as you have gives comment like to make a big bubble on the surface, like the graphene itself cover all this area, okay, and this cover whole area, the graphene sheet, and then simultaneously bind the other things. But as per your given comment, you suggested that this part is covered by whole graphene. So we have already work on it, and we give commands to Jaya that she will make the whole cover the whole surface area with the graphene sheet, and then accordingly the other things will combine on the surface. And also, we, as per your given comment, we modify this figure as well. We go with the nasal pharyngeal swab. So we just take this kind of we modify this picture as well, and as per your given comment, and this is a schematic representation of the chemical process. So as per your given comment that you suggested, that also mention here the chemical structure as well, like the chemical structure of the graphene sheet, and what is the chemical change if we put the ADCNHS here, and then afterwards the if we combine with the antibody, so what is the changes in terms of the chemical bonds. So we have to mention here in this section, and simultaneously also what is the, there is no such as recombinant protein combined with the antibody, and then what is the basic changes in the chemical bonds. So we have to mention here as per your given comment. So this is our result that it's a sensitivity capacitance IDC results. So we have the different kind of concentration. So we have 10 concentration, and then we measure the consecutive each steps. It's basic things that we have followed and or we have mentioned in the paper as well. It's a sensitivity detection and the selectivity as well. But as per your given comment that we have to deleted this part is a PSA and AD plasma. So we have already deleted this part, and also change in the content in the paper content as also. So we have to skip this part, and we just focus only this SARS-CoV-2 antigen. As per your given comment, we already work on it and change in the figure as well as in the content also. Sir, we have prepared the sample for the SEM measurement. For the SEM measurement, for like the antigen picture we don't have, so we already prepared the sample. And also, as per your given comment, you said that also make one another chip after the washing of antigen, and then again to check the SEM, because we have to prove the reusability factor. So the two chip we have to prepare for this SEM measurement, as well as this AFM image also, like we have to measure antibody picture and antigen picture also. And one is another, after washing this whole cycle on the chip, to we have to wash again, and then what picture comes up with the bare with the uh, piranha cleaning. So what picture it comes up with the after the AFM. So we have three different kind of samples we have to measure, in this case AFM, and two for SEM. And this is the FTIR figure. So as we can see that there's a change in each peak. Black one is having the bare, and the blue one is having the graphene oxide, and the red one is having EDC-NHS. It's a third step, and the fourth one is having the antibody. So you can see that there's a linear changes, so in each bond having the different kind of changes. So we defined well here in the case of FTIR. And I can skip this one. So this is the water contact angle measurement. So we did the water contact angle measurement as a reference, and then graphene oxide is having 80.4, and then EDC-NHS 75.4, because the left angle is quite wide, and that's why we got, because of the viscosity of this liquid. Why we are doing water contact? Because to measure the viscosity of the liquid, and we have clarify in terms of the angle of the drop and the surface. In these two cases we have to measure the angle, water contact angle. And so we have already make the sample for the another two step, like antibody and antigen. But I don't think so we have to go further in the water contact measure. So we have to just mention only these three, and we put in the paper only three this thing. And sir, we have finalized until EDC-NHS, and also we finalize the antibody. Up to antibody we mobilize today, and tomorrow in front of you we mobilize antigen, and we will show you that the value become changes in each concentration of antigen. And here we also mentioned the video that when we mobilize the antibody,
we are having SARS coronavirus spiked antibody. So now we are going to mobilize on EDC-NHS. So here we have a spiked antibody. So now we mix it well. So this is our chip. Now you can see.
so here we immobilize antibody after the EDC step, and tomorrow we will immobilize the antigen in different kind of concentration. So we have the 10 different kind of concentration of antigen sample, and in front of you we use this chip and to mobilize antigen. And then we have check the different kind of capacitance ranges in terms of concentration.
Yeah.
Thank you, sir.